Welcome to today's webinar, Immunosequencing Analysis and its Clinical Applications. I'm Nico Cavello, Product Marketing Associate at GeneWiz, and I am very pleased to introduce today's presenter, Dr. Antonina Silkov. Dr. Silkov completed her PhD at Weizmann Institute of Science, where she studied transcriptional regulation of apoptosis and immune response. She has 16 years of experience in bioinformatics, and her primary interest is analysis of sequence and structure data related to development of cancer and other diseases. Dr. Silkov is currently a bioinformatics analyst at GeneWiz, focusing on immunosequencing analysis. If you have any questions for Dr. Silkov during the webinar, please enter them into the Q&A section, and we will address as many questions as possible at the end of the session. Without further ado, please welcome Dr. Antonina Silkov.
Thank you, Nico, for your introduction. I will start today's webinar with describing structure and function of B and T cell receptors. Then we'll talk about generation of antigen receptor diversity that is required for adequate immune response. We'll go through the most widely used types of immunosequencing analysis and finally application of this analysis in antibody discovery. B and T cells or lymphocytes are the major players of adaptive immune system. Their receptors are responsible for the pathogen recognition and play a crucial role in immune response. As you can see here, B and T cells are heteromers, which means that they consist of different parts. B cell receptors have light chains and heavy chains, and T cell receptors have alpha chain and beta chain. B and T cell receptors are cell-bound, but B cell receptors also have a soluble form called antibody. B and T cell repertoires are highly diverse. Such diversity is made possible by several mechanisms. The first one is combinatorial joining. Heavy chains of BCRs or B cell receptors and beta chains of TCRs are composed of three segments called variable, diversity, and joining regions. Light or alpha chains are composed of two segments, V and J. In the germline DNA, the variable, diversity, and joining regions are encoded by numerous discontinuous genes, and only one gene out of the group will become part of the mature lymphocyte. So this is the first step in generating diversity. During the journey, nucleotides are often randomly inserted or lost at the ends of recombining fragments. This process is called junctional diversification. Rearranged heavy and light chains or alpha and beta chains can be paired in different ways, and this is another source of lymphocyte receptor diversity. Finally, there is another diversification step which is specific to B cells, and this step is following antigen exposure. The process is called somatic hypermutation. Through the variable regions, mutations are introduced such that a modified receptor can have increased antigen affinity. Here you see the representations of B cell receptors, BCRs, and T cell receptors, TCRs, consisting of uniform structural blocks. Each block is represented by an immunoglobulin or Ig domain. Ig domains consist of antiparallel beta sheets. Because of structural requirements, the sequence of these sheets are quite conserved. On the contrary, the loop on one of the ends of Ig domain are quite flexible and their sequence is variable. So these three loops constitute so-called complementarity determining regions 1, 2, and 3 that are responsible for antigen recognition. The diversity of lymphocyte receptors can be studied with the help of next-generation sequencing or NGS technology that have rapidly developed over the recent years. Both genomic DNA and mRNA can be used for immunosequencing analysis. Sequences of genomic DNA allows estimation of the clonality of the given sequence, in other words, the number of lymphocytes expressing the receptor or antibody. Using mRNA as a template can provide an estimate of the relative expression level of the sequences in the repertoire. The bioinformatics analysis consists of two parts. First, assignment of the germline V, D, and J regions and CDR regions, and second, the data analysis of itself. So these are the main types of data analysis or immunosequencing analysis. Most of the types of analysis are common to B and T cell receptors, whereas the last three types are specific to B cells because they handle somatic hypermutation. Because of a time limitation, I will talk today only about the most common types of analysis, which include CDR3 length distribution, VDJ usage, clonotype frequency, comparative repertoire analysis, and phylogenetic analysis. CDR3 length distribution. CDR3 is the most variable region of the immunoreceptors because this region lies on the junction of variable diversity and joining regions, and also because in this region is subject to nucleotide diversification. The distribution of the CDR3 length is bell-shaped in normal individuals. You can see it on the left panels on the slides, where you see the healthy adult on children cohorts. On the right panel, you see the disease cohorts that have very severe aplastic anemia. This is the bone marrow dysfunction that results in the reduction of the number of white and red cells, or hepatitis-associated aplastic anemia. You can see that in disease cohorts, the distribution is skewed and the maximum is shifted towards the longer CDR3 sequences. Of course, with sequencing, one may obtain more detailed information than just a regional length. The next analysis we are going to discuss is identifying the germline genes involved in immunoglobulin rearrangement. The methods for VDJ assignment can be divided into two main classes. One is alignment-based and another is model-based. Here you see the examples of applications of this analysis. One is the search for the origin of malignancies in the post-transplant patients that often develop lymphoproliferative disorder. Sometimes they have multiple malignant clones, but sometimes the clone originates from the same cell. Another application is detecting disease-related VDJ combinations. For example, V delta 2 J alpha TCR rearrangement is frequent in precursor B acute lymphoblastic leukemia, but practically absent from the healthy individuals. Clonotype frequency. This is the exact sequence of lymphocyte variable region. Upon recognition of the specific antigens, lymphocytes can undergo clonal expansion. The clonal distribution and diversity of lymphocyte repertoires may represent a fingerprint of immunological status. For example, on this slide, you can see that lymphocyte repertoire 1 represents a uniform repertoire that most likely corresponds to a healthy individual. Lymphocyte repertoire 2 is dominated by red clones that most likely underwent a large expansion. And this means that this clone dominates the repertoire, which is the result of disease, infection, or vaccination. Cutaneous T-cell lymphomas are a heterogeneous collection of non-Hodgkin's lymphomas. Early diagnosis of these diseases can be challenging. because skin lesions of some CTCLs can clinically resemble those of benign inflammatory disorders, including psoriasis and atopic dermatitis. In 2 patients shown on this slide, analysis of next-generation sequencing demonstrated clear malignant clones. With the progress of NGS, various disease-specific databases of lymphocyte receptor sequences became available. This prompted researchers to look for the common disease-specific motifs in the antigen-recognizing regions of TCRs and BCRs of individuals with the same disease. Such motifs could become important diagnostic markers. There are multiple examples of convergent repertoire sequences. Some of the examples are listed in the table here. Convergent CD3 antibody sequences are observed in the patients with dengue infection, after Haemophilus influenza and tetanus toxoid vaccination, and in patients with celiac disease. However, the convergent repertoires have been found only in a limited number of diseases. One example illustrating a limited disease-specific repertoire convergence is analysis of identical twins vaccinated with varicella zosters. Researchers found that twins had a larger number of shared clones than unrelated individuals, but their immune response was largely dominated by unique B-cell clones. Detection of HIV broadly neutralizing antibodies is an example where a simple sequence comparison to public repertoire is inefficient and a phylogenetic analysis is helpful. Approximately 20% of HIV-infected individuals develop broadly neutralizing antibodies that can be efficient against a wide variety of HIV strains. In 2010, a new class of such antibodies called VRC01 was discovered. It neutralized more than 90% of HIV-1 strains. Its broad spectrum of neutralization is based on its ability to neutralize a conserved region on the viral epitope. HIV infects cells through spikes that are on the outside of the virus. The tip of the spike is formed by glycoprotein 120, GP120, and this sticks to the part of immune cell called CD4. VRC01 binds to GP120 and disrupts interaction between a virus and lymphocyte. VRC01 antibodies have characteristic properties that include heavy chain origin from the same germline gene, light chain characterized by the CDR3 region of 5 amino acid, and a long or protruding heavy chain third complementarity determining regions. A group of scientists from NIH and Columbia University selected an HIV-infected donor called here C38, whose sera exhibited broad and potent neutralization of HIV. They performed NGS analysis of antibodies from this donor, and they compared these sequences to the known VRC01 antibodies. The sequence comparison produced no results. Then the researchers decided to perform cross-donor phylogenetic analysis of heavy chains, and in the analysis they included all the sequences from the donor C38 and known VRC1 antibodies. And they focused on the part of phylogenetic tree that included both types of sequences, donor ones and known VRC1 sequences. Then they selected several antibodies from these branches and tested them for neutralization. And indeed, they detected antibodies that neutralized HIV. So they concluded that this method helps identifying VRC1 antibody. This identification is of course useful in diagnostics. The VRC01 libraries can be also used in vaccine development. The researchers from the Scripps Institute adopted a vaccine priming strategy which consists of activating B-cell precursors with antigen. In the first step, they engineered germline reverted antibodies that contained germline VJ genes and mature CDR3 regions. So they tested interactions of these antibodies with antigen GP120. Unfortunately, they found no interaction, so they used the libraries, both VRC1 antibodies and reverted germline libraries, to improve the property of GP120 protein by mutagenesis. So they tested the mutated versions, how they bound to the antibodies, and finally they obtained the engineered out domain with improved binding properties. Then they used this domain to screen the B cells of uninfected individuals, and they sequenced the positive B cells, and they searched for VRC01 class antibodies. And they did identify the VRC1 class precursor, which according to their estimation is found in 96% of individuals with certain germline alleles. Of course, such a precursor is a very attractive target for vaccination. Until now, we discussed the application of immunosequencing analysis in diagnosis of disease. However, applications include also prognosis and monitoring not only the disease states, but the states of aging and vaccination. Immunosequencing analysis is useful in vaccine development, which we have already discussed, and antibody production, transplantation donor-recipient matching protocols, and development of immunomodulatory drugs. I would like to focus on antibody production that represent the fastest growing segment of biotech industry. Currently, two most popular platforms of antibody discovery are hybridoma technology and phage display. In hybridoma technology, a mouse is immunized with antigen. Its spleen cells are collected and fused to myeloma cells that makes them immortal. The two most popular platforms for antibody discovery now are hybridoma technology and phage display. In hybridoma technology, a mouse is immunized with antigen. Its spleen cells are collected and fused with myeloma cells. This makes cells immortal, and hybridomas secrete, hopefully, antigen-specific antibodies. The cells are diluted so that one clone secretes a specific type of antibody, and the antibodies are screened and collected. This technique has certain limitations that includes species restriction due to myeloma requirement. Only mouse cells can be fused to myeloma. Inefficient fusion, loss of positive clones, and this technique is labor intensive. In phage display, the antibody library is displayed on the surface of phages, and then the phages undergo multiple rounds of panning, elution, and amplification, and this leads to enrichment of the phages expressing antibody-specific antibodies. After screening and cloning, the antibodies are collected. However, this technique suffers from expression bias, increased off-target binding, and finally, antibodies produced by this technique have lower affinity than natural antibody, and this is because these antibodies do not undergo the process of somatic hypermutation like in the germinal centers of immunized animals. Antibody discovery helps overcoming these caveats. For example, the starting material can be any species. Next generation sequence can capture the entire repertoire. The sequence selection is performed using immunosequencing analysis, and it significantly reduced the clones chosen for the further selection. And this makes the techniques fast and less labor intensive. The immunosequencing can be used in conjunction with conventional methods. For example, NGS screening of hybridomas can prevent the loss of positive clones. The NGS screening of libraries can determine their quality, which includes library size and diversity. NGS during the stage of panning and amplification can determine the enrichment factor, which is used for selection. This slide illustrates the utility of immunosequencing in generating anti-RON antibodies. RON is a receptor tyrosine kinase that mediates neoplastic transformation. Aberrant expression of RON leads to activation of common cancer pathways and finally leads to proliferation and survival, migration and invasion, angiogenesis, and chemoresistance. Currently, RON-specific therapeutic monoclonal antibodies are being tested in preclinical models and clinical trials, for example, in pancreatic cancer. A recent study from the Belgian group used NGS to make the discovery of functional antibodies more efficient. A candidate immune library directed to the RON was first displayed. The immunosequencing analysis was applied to antibody selection process. The sequences of antibodies were clustered, and cluster size was found to be an important parameter for selection of RON-specific antibodies. Also, authors calculated the enrichment factor of the antibodies as compared to negative control. So you can see that these two parameters could select a large varieties of antibodies, and blue square correspond to the clusters that have been already identified by conventional methods. Out of the new clusters, a series of clusters were randomly chosen for further evaluations. And 90% of these clusters were shown to be binders, RON binders, and 45 were efficient in ERK phosphorylation assay. So the conclusions of my talk is that immunosequencing is a powerful tool for estimating repertoire diversity, for linking lymphocyte repertoires to antigen specificities. And this analysis has a great potential in the clinical applications that includes diagnostics, antibody discovery, and vaccine development. Thank you for your attention, and I'm ready to take your questions.
Thank you, Dr. Silkov. I appreciate you taking your time. Sorry for the technical difficulties. One of the first questions we got is, how are the complementary determining regions identified?
As I said, complementarity determining regions are much more variable than the framework regions. So research is looking for the conserved sequences that can be extracted from BCR and TCR databases. One of the most popular databases related to the system is IMGT, although nowadays other databases has always also been developing. Thank you.
The second question is, does isotype class switching contribute to antigen receptor diversity?
Isotype switching occurs at the constant region of the heavy chains of B-cell receptors. Because it does not involve variable region, it does not affect antigen binding specificity. However, it's a very important process, and many cells of immune system, like natural killer cells and macrophages, bind the constant regions of the antibodies that in turn interact with antigen. So the composition of the constant region determines the subsequent immune response.
Thank you, Dr. Silkov. The third question is, how can we distinguish between true receptor repertoire diversity and PCR and sequencing errors?
This is an excellent question, and this is a big problem in interpreting NGS results and differentiating between false and true diversity. And different approaches have been developed One of the simplest one is abundance-based. For example, one can eliminate low abundance sequences that differ from high abundance sequences by one nucleotide. Another also simple approach is removing low quality sequences. However, in this case, one can probably lose a lot of information. So now people are developing more complicated approaches, for example, alignment and merging of low quality reads with high quality reads. So this approach was shown to be very efficient in discriminating between true and false diversity.
Thank you, Dr. Sokov. So this is all the time we have for today. I believe we got through all of the questions. I would just like to thank you for presenting as part of our GENEWIZ Week webinar series. Again, I'd like to apologize to the audience as well as Dr. Sokov for the technical difficulties. As mentioned, we will upload these slides in the presentation and share them with all the attendees so they can review the full webinar. Please make sure you come to our final webinar session, which will be held tomorrow, Friday, June 16th at 11 a.m. With Dr. Yixin Wang, the Chief Scientific Officer at Celsea Diagnostics. Thank you very much for attending today's webinar and have a good day.